A very warm welcome to the participants of the Proteomics Advanced Winter School 2021. Presented by Proteomics Lab, Indian Institute of Technology, Bombay. Day 2, Sample Preparation and Gel-Based Proteomics. Lecture 1, Sample Preparation.
This is quite busy slide, but it shows you that when we talk about a complex task, something like looking at proteome analysis, which means we are looking at a large number of proteins to be analyzed, and potentially all the proteins to be analyzed from a given organism or given organ or given cell, then I think we have to come up with different extraction strategies. So the first circle here shows that whether you can have a plant, you can have a cell, you can have any type of human sample, any other bacteria, any variety of sample type. You first want to come up with novel strategies for protein extraction. And once you have extracted protein and you have removed other contaminants, which is essentially nucleic acids, lipids, carbohydrates, salt, then you want to separate the proteins. And for protein separation, which is the next step, then you have electrophoretic methods like gel-based proteomics approaches, including the one-dimensional LDS or two-dimensional electrophoresis or difference in electrophoresis type of technologies. Or you also have gel-free methods, primarily mass spectrometry based methods, which are isotope coded affinity tag, multiple dimension protein identification technology, or various type of label based methods like tandem mass tag or I-track or isobaric tag for relative and absolute quantification. So again, after you have done protein extraction, you want to separate the protein and also you want to quantify the protein, and that's where a lot of things happens at this stage. And then, of course, in the process, you want to identify what these proteins are, which are looking interesting to you. Either they're coming from the gels or coming after labeling the sample from the different labeled strategy like ICAT or I-track. And then you want to identify them using mass spectrometry platforms, whether it is multi-top or ESI QTOF or Orbitraps. And then you also want to do a lot of bioinformatics work to search the databases to find out where is your protein located, and is that you are looking at the right protein. So again, database search and bioinformatics becomes very crucial for any kind of big data field, but more so for proteomics. Once you have done protein identification, now you know the set of proteins which you are maximum interested in, and you want to investigate them further to understand a given biological system. Then you want to move on to the protein characterization part. And that's where different type of functional proteomics technologies like protein microarrays, different type of label-free biosensors like surface plasmon resonance, and other label-free biosensors are available, which are helpful for protein characterization. And now once you have narrowed down some of these leads and you are more keen into a very specific protein study, then you can look into the conventional organic chemistry and conventional chemistry techniques using the protein purification methods, using the X-ray crystallography, and do there more of the structural investigation. So I hope you are now slightly understanding the broader picture that for looking at the complex proteome, we have to do the series of experiments, and we have a variety of choice in the process of doing these proteomics based investigation. But the first part becomes most crucial because there are too many steps and too many technologies which are linked later on. But to begin with, you have to start with the good protein extraction methods. And that's where I think, Dr. Jingre in the beginning of the inaugural talk mentioned in discussion about experimental design. And I thought to bring that point first up front, that your sample preparation strategies are many times linked to the experimental design. For instance, I have just shown one circle here talking about gel-based approaches. Let's say you are interested to take your protein extract and separate them in the electrophoretic method, and you want to separate them either in one dimension with SDS page, or you're looking at two-dimensional electrophoresis based method. Your sample preparation strategy will slightly change because the buffers which you are going to use, they are going to be more compatible for the electrophoretic methods. And most of the time you're going to use urea, thiourea, chaps, rehydration solutions, which can contain these things along with bromophenol blue, which can help you to track the progression of the protein movement in the gels for SDS page or 2D gels. But when you're looking at more quantitative approaches like two-dimensional difference in the electrophoresis or 2D dyes, then your experimental strategy again changes, because now you have to think about you need sufficient amount of protein so that your control and your test, they are going to have not only the sufficient amount of protein for running on the gel, but also you should make some internal standard, which I have labeled as IS here, that you need to make the pool of internal standard, which has to be labeled with another dye. And as we go in the the next modules tomorrow about gels, we will talk about what these dyes are of Psi 3, Psi 5, and Psi 2 dyes. But here I'm just giving you an idea that you have to also think about which technology you are going to use to address your question. If the goal was to look at only 2D gel, you're going to look at very huge amount of protein, because each of the gel which you're going to separate on the 2D, you have to run them separately. And the dyes which you're going to use are primarily going to be Kumasi blue or going to be silver stain sometime. But still you need at least 250 to 500 microgram protein, and preferably even 1 milligram protein, which is of high quality. For doing dyes experiment, you can have even 150 microgram or 200 microgram protein, and even that is sufficient. But you have to keep in mind that how many internal standards you have to make. Accordingly, your experimental design will change. Now come to mass spectrometry. For instance, you're thinking about going directly with the shotgun proteomics approach, and you want to perform a label-free mass spectrometry based technology. So now you would like to think about, in my experiment, I should have a sufficient amount of protein. Let's say you want to compare 50 control and 50 disease samples. So first you should think about, like, you need to have sufficient amount of protein to digest for peptides. And then you should also have sufficient amount of protein from each of the sample so that you can make a small pool of that. And that pool has to be made sufficient quantity so that periodically you are going to run them in the LC-MS. Let's say after every 3 sample you want to run one of your pool sample, which you are going to monitor over the time period, because the LC-MS runs takes time. Every sample is going to run 2 or 3 hours, followed by same time for the running the your blanks. So in every day you're hardly going to run 4 or 5 samples. And if you want to compare now 100 sample, you have to go a couple of weeks time to just run one project. So given the changes happen and so much reproducibility issues comes in any technology, it's good idea to monitor the reproducibility of the instrument and also the intensities of these peptides so that you can use that for normalizing over the time period. So again, think about experimental design and strategies. That's very important, because your amount of protein which you want to extract from any sample type, you have to think these things ahead of time, that how much protein you need. And in this case, when you're looking at label-free quantification, you also think about what is going to be your internal standard, which you might want to spike in along with your sample of interest. Or sometimes people also use the retention time peptides, which is IRT peptides, so that you can monitor the chromatography, how reproducible it is coming on a specific time when the peptides are diluted out from the LC-MS column. So again, many of these things may not be very clear to you right now, but it will become more and more apparent as we keep going in more detail about each of these technology, and we talk about how that works. But I'm just trying to give you idea that it looks very interesting to think about. I want to do proteomics, I want to do quantitative proteomics, and I want to perform any of these experiments, but we have no idea that how crucial the experimental design is going to be, how crucial the sample preparation is going to be, and how much thoughts we should give in beforehand that is going to determine the success of our experiment. So for instance, now when we look into label-based mass spectrometry or quantitative proteomics approach using labels like iTrack or TMT. So iTrack and TMT, they both bind to the peptides on the N-terminal sequence, and you label them with these reporter ion-based the tags, which is tandem mass tag or isobaric tag for relative and absolute quantification. So once you want to quantify your samples, you have to think about again, how are you going to analyze the data? How are you going to ensure the reproducibility of the data? So a simple experiment for this type of workflow could be, think about a 4-plex iTrack experiment, right? When you have 114, 115, 116, 117, these four reporter ions tags, which you want to label your four conditions. So you have a control and you got, a time course experiment, the sample taken out after 2 hours, after 6 hours, after 24 hours. And now you want to compare all that with the control condition or zero time point. So now you are looking at labeling all of those four with the reporter ions from 114 to 117, and you'd like to now compare them across. So while doing so, this is your one replicate. Now you have multiple replicates which you want to compare, and more your conditions are going to be increasing. So now when you want to cross compare your one labeling reaction with other labeling reaction, again you have to think about how are you going to compare the intensities of one reaction versus other reaction, and how to cross compare that. That's where making a pool which you can use in one channel is very important. So in this strategy, if you are looking at 114 to 117 4-plex only, you have to make one of the channel as the common reference pool, which you are always going to have a common sample which you are going to run, and that common sample you are going to monitor the intensity of that reporter run of 114 in multiple conditions, so that you can cross compare your different quantification of different conditions from one reaction to other reaction. So I don't want to afraid you that we are talking about many technologies right now, but I only want to convey you one single point, that there are many technologies in the proteomics field, whether you talk about gel-based approach, electrophoretic methods, or you talk about mass spectrometry-based methods, which is either label-free or label-based. In all of these, the considerations are on the experimental design, which is very crucial. Otherwise, you will do so much hard work, a lot of data will be generated, but you can't make it publishable, you can't make it of the conclusive quality. So when we talk about the sample preparation, given the audience who is all part of this workshop, we have very heterogeneous group of people who are coming from variety of background, and we have to think about every biological sample brings their own specific type of challenge, very much uniqueness from that sample type. For example, plant cells, they're very harsh tissue, they have a lot of interfering substances. Plant leaf will have ribisco type of abundant proteins. Plant roots will have polyphenols and many other components. Many plant cells have a lot of pigments and colors. All those are going to be interference, which we have to remove if you want to prepare that sample to do the proteomic analysis. Fungal cells, again, is very tough sample to work with. Lot of proteolysis challenges are there. Bacterial cells have very high ratio of nucleic acid, which you want to remove before you move on to your proteomics work. When you want to look into clinical samples like cerebrospinal fluid, is very dilute sample which is having very high salt concentration. You want to first remove that, and also there is a trace of lipids which you want to delipidate. Then only your samples are ready. Your biological sample, which is very commonly used, is serum or plasma, which contains abundant proteins like immunoglobulins, albumin, and salt. You have to remove all of these before your sample can be used. You are doing a lot of tissue culture based experiment. For growing those, you are using the buffer, you're using the salt, you're using the serum, and all those are coming as a part of your sample, which you have to get rid of for doing proteomics. So I hope you got some idea that there are different type of samples, and I've just listed a few. I'm sure you can come across with your own sample type, which you are most keen to work with, and you will yourself understand that there are challenges of handling that sample. And this is where I think a user is best person who knows about their biological questions and biological sample. And you have to be intellectual enough to understand that what are the interferences which are going to come on your way if you want to prepare very clean and good protein sample, because everything else has to be removed from that sample except protein to take it forward. So let me focus a little bit more on the clinical proteomics part. That is something I think large number of participants are from clinical or veterinary background, and even we do a lot of work in this area. So when you are planning for any kind of clinical proteomics based work, even prior to starting your study, as I said, there are two things. One is what is your biological question, what technique you are going to use. That is first thing to decide that which technique you are going to be using, and how best you can address that with your sample preparation strategy. But also, when you are planning to collect your sample, you have to keep in mind that there are many analytical variability are there, and you want to first look at what could be pre-analytical variability as well. So for instance, many factors which are going to affect your sample collection and sample storage, those are all pre-analytical factors. Even before you start your project, you are going to collect many samples, and the samples are going to be, having the challenges of how best they were collected and how best they were stored. So there are too many factors over there only for thinking about this aspect. And there are, of course, many factors, some of those which we have no control, like intrinsic influences of gender, age, ethnicity, etc. And extrinsic influence also, like, your participants who are going to be enrolled in the study, what type of diet they have, what medication they take, what is their, smoking or non-smoking behavior, alcohol consumption behavior. So again, many of these things should be part of your clinical data record forms, where the participant, if they can disclose these information, that could be very informative, because you are, let's say, looking into something for metabolomics and analysis, and a set of participants are consuming alcohol, and then you are very happy that you see a lot of set of metabolites are very changing in those individuals, but those may not be influence of a given disease if you are investigating that, but could be just because of the intake of certain specific type of food behavior, right? So that's something which we have to keep in mind. If possible, as detailed clinical record forms or study design forms are made, that can minimize lot of issues at the end when you're analyzing the data, because you'll always find there are some outliers. There are some individuals which are showing variability, and you have no idea, is that biological or is that because of anything unique to that individual, right? So that's where you have to keep many of these issues. Of course, there are many technical pre-analytical factors. For instance, how you are going to collect the sample, process the sample, and store the sample, because many a times you are handling the sample, which are biofluids, which are going to be degraded very fast if you do not collect them properly, do not process them quickly, and do not store them in good -20 or -80 type of conditions. Even sometimes the sample collection mode, like the different type of clinical sample, what is the posture of the patient when they are collecting the sample, uh, or even something as simple which we even do not pay attention, that what type of containers and tubes which you are using for sample collection. You might be using, based on availability of the stock of the tube, one set of sample of 50 patients you collected in one tube type, and that tube stock got finished, and now the next batch of the individual which are supposed to be controls, now you are collecting in a different type of tube, and now you are going to compare them for proteomics investigation, and you see, wow, big change in my control versus disease. And that's all coming because of the type of tube, and not anything specific, what the biological questions you want to address. Again, many of these things can be confounding, and these are very simple things, but these are very crucial at the time of sample collection. So it's a good idea to make some sort of your chart to think about do's and don'ts, and think about, if you are planning for a bigger study, how best you can get the reagents and resource which is sufficient for that whole project, so that you don't have to keep changing your things in between as you go along. Otherwise, you'll have the problem of lot of reproducibility issues, which is not biological, but because it is of the pre-analytical factors which are creating those kind of variability. Many times, a lot of tubes, even the plastic quality is bad, and you will see, those components are getting shedding in the directly in your sample. When you are processing that in the mass spec, and you can see those traces, and those can be very confounding. Even people have shown some study of the cerebrospinal fluid measurements when they look at beta amyloid and tau proteins, and they found that they differ when they collected in the tubes of different type of material. So same sample, when you collect in different type of tube, the behavior, the what you measure for beta amyloid and tau proteins, which are aggregating very fast, you are going to see them very different. These are some experimental type of work which has shown that why it is so important to pay attention to a small thing like the quality of your tubes, the storage conditions, right? And again, I'm sure all of you are quite experienced in the field, but you have to keep in mind that multiple freeze thaw is going to be very detrimental for any sample. So while you are collecting the sample, it's a good idea to make multiple aliquots of the sample, so that those samples could be stored in different boxes, and you are going to only handle few boxes at a time, and not you are going to change all the time your sample, and everything is getting thawed. So these are some small tips, but I'm sure these are going to be useful when you are planning for a bigger experiment. And there's a lot of investment which goes on to do proteomics or any other omics based experiment. But many times you are introducing a lot of variability at the pre-analytical level itself, which no technology can actually try to correct that part. Because I was talking to you about thinking about experimental design and add some sort of internal standard, add certain pools, look for more type of thing which can monitor the chromatography reproducibility. And all of those are a way to actually ensure that there is no analytical variability, and you are trying to normalize those, you are trying to combat those type of variability. But how are you going to change these things? Like, think about some serum samples, some technicians, they have collected and they kept it on the room temperature for 5-6 hours, which is very common. And some samples were immediately kept in 4 degrees, some samples are kept in -20. Eventually you got a stock of 100 samples which contains all the 3 types. All of them have already got a lot of variability to start with, and you are not going to at the end going to find anything which is very reproducible, because you have lost too many things in the statistical analysis while considering all these samples from the same type of patients. So I think your clinical collaborators, your student staff, your collaborating team who is working on these projects has to be very meticulous, because even a small sample type could be 20-25 sample, but very well collected, very well designed, could be highly powerful if you do it thoughtfully. And please take it seriously that there's a lot of investment you are going to put in, lot of time investment you are going to put in for analyzing the data and trying to make sense of the data, but that will not make any sense if these parts are not done carefully. The first 2 part, what is your biological question to be addressed, for which technique you are going to take, which platform you are going to use, what will be the strategies in that platform. Second is your input sample type, how well it has been collected and how well you have removed the variability. Again, just for a practical experience, what we try to do for any project when we start something of the clinical project, we do try to buy the reagents in bulk. We do try to buy the plasticware in bulk and try to provide to our clinical collaborators the same reagents, so that at least that much variability can be reduced. We also do try to optimize the conditions in our lab here and try to first mimic that in the collaborating hospitals, then only the samples can be collected. But again, despite all that, you will have still some variability which you cannot avoid given the nature of the samples, and you're talking about the patient samples, you're talking about the people which you have no control on, and you will have some degradation and stuff, but you have to avoid that as much as possible. Also, like if you're looking at clinical samples, it's a good idea to reach out to the protein extraction and peptide preparation as quick as fast as possible, because your raw sample, crude sample which you are storing, whether it is serum or cerebrospinal fluid or tissue or any sample, is going to degrade. Whatever you do, your box is not open, but your colleague is going to open the freezer multiple times, and the freeze thaw will happen at least partially, and you will see progressive degradation of the sample stored for a long time. So it's good idea to, when you are collecting the samples fresh, good idea to process them and try to get the protein, followed by digest them to get the peptides. And all of that is ready for you to store them in your -80 freezers. Those will be highly stable and high quality sample which you can analyze over the month's time. So with this, I hope you have conveyed you 2 important points, that proteomics is not one technique and not one workflow. You have to think about which question, which technique to take forward. And second, as carefully you can do your sample collection and storage, that will be highly beneficial for your next set of activities. So coming to the sample preparation in proteomics, we are talking about variety of things here, whether it's we are looking at whole yeast proteome, whole organism, or looking at only specific type of tissue. We work on sometimes brain tumor tissue, or look at any specific fluid like serum, plasma, CSF, urine, saliva, nasopharyngeal, so a variety of samples, or looking at different type of cell-based work. In all of them, sample preparation is crucial, most important, and very different. Very different type of strategies has to be taken forward, and as reproducible you can prepare these samples from different replicates, more and more meaningful data you will get from the technology which you're going to employ later on. So this workflow is going to involve multiple steps and not in the same order for every single sample type which I showed you, but you are going to primarily look into how best to lyse your cell or disrupt the cell, how to protect them from proteolysis, how well you can fractionate the complex sample to make it simplified fractions from so that you can get the better sample output from those, protein extraction and solubilization strategies, removing the contaminants. Contaminants could be many things, your abundant protein to the salt to the pigments, or for that matter, nucleic acid, lipid, carbohydrate, anything is contaminant other than protein, and you have to think about carefully how to make sure that those things are minimized or removed. And then of course protein quantification before you are ready for your next set of steps. So often you will use different type of precipitation procedures in proteomics workflow. Acetone is commonly used when you can simply use 3 or 4 volume of your ice cold acetone, and which is really chilled at -20, and then you allow that for the protein precipitation with 1 to 2 hours of incubation. You do the centrifugation and remove the acetone, and that is quite a quick and efficient way of removing a lot of lipids and detergents which are present in your sample, and then you can proceed for that type of sample for further analysis. Or you can use TCA, trichloroacetic acid, for precipitation, very effective protein precipitant, and 10% TCA is quite efficient for precipitating the protein. If you incubate that in ice for 30 minutes, then you are going to get the protein pellet, which you can wash with acetone and ethanol, and you are going to have good sample recovery, so you will not lose much sample with this type of precipitation method. Often people use protein precipitation with TCA and acetone together. So now you weigh the TCA with weight, and then put that in acetone in solution form, make that mix, and then you are going to lyse your cells in 10% TCA with acetone, which also contains the dithiotritol to break open the disulfide bonds. And then you are going to incubate that for 1 or 2 hours in -20 degrees. Then you are going to wash your pellet to remove the TCA, and you can wash with acetone alone, which contains the DTT. And in this manner, now your protein pellet is pretty clean, and that is very commonly used strategy for sample preparation. So just to summarize this part, I think you will often see, again, if you are not sure about what type of sample prep you should be using, you should try out some of these methods yourself and see, like, after extracting the protein from each method, when you run them on the gel, what kind of quality of the protein you can see. When you quantify the protein, what amount of quantity you can get from these type of strategies. And this is how you are going to develop a protocol. So protocol development from any sample type should be explored with variety of methods which you should be exploring simultaneously. Over the time period, what we have found that various type of projects, especially cancer projects, they require tissue-based investigation, and given the complexity of the sample and tumor tissue, followed by you want to minimize the steps required before you can perform the mass spectrometry analysis. I think urea lysis method is one of the efficient way of moving forward. And here what you do, like you have the fresh frozen tissue, which you always want to go as minimum as possible. In our personal experience, you can go even as low as 5 milligram of tissue, which is properly frozen and snap frozen in liquid nitrogen. But if you can have 20 to 25 milligram minimum, that will be ideal situation. Of course, if you have 100 milligram, that will be really good to do sufficient protein extraction and store it for the future as well. But even if for 1 or 2 experiment, as long as you got, many times we get the the sample from the needle biopsy, very small amount of tissue, but even that is possible to do as long as we can get even 4 to 5 milligram of tissue to start with. Now in that you can add the urea lysis buffer, which contains 8 molar urea, tris buffer, sodium chloride, magnesium chloride. Then homogenize the tissue with the sonicator and bead beating, followed by you can do centrifugation step, and then you can collect the supernatant which is having your protein, and that can be taken forward for the next steps of quality control check. We had this workshop last year, the DST workshop only, where John Wilson from Profiti, he came from New York, and he was involved in the sample prep strategies workshop part. And I know he made a very nice comment that this entire procedure of sample preparation should be carried out in ice. It should be cold and fast, which are the cardinal rule of proteomics. So of course, when you are working on the proteins, you have to try to keep the things really tight. Everything should be prepared in advance, and try to do everything in the cooling conditions so that you can minimize all the degradation. Otherwise, those will be difficult for you to overcome in the analysis stage. Let me show you this brief hands-on demo activity of urea lysis method, how to prepare that.
Hello everyone. So for the success of any proteomic experiment, sample preparation is the most crucial step. Sample preparation involves protein extraction, protein digestion, sample cleanup, and final mass spectrometry. Depending upon the experimental objective and type of biological sample, different protein extraction protocol can be used. Different biological samples could be bacterial pellets, cell pellets, or tissue sample. Today we will demonstrate you the protein extraction method for a tissue sample. Here we have already weighed amount of tissue. For this we will use urea lysis buffer, which is 8 molar urea. Appropriate volume of this buffer is added to the tissue. It is vortex for a few seconds and spin. After addition of buffer, the next step is homogenization, which ensures that tissue is completely light in the given buffer. So for this, now we will use sonication. For this, we are using probe sonicator. Whole procedure of sonication should be performed on ice. So after this sonication, whole of the protein will be extracted into the buffer. After sonication, it should be made sure that whole content is uniform and nothing is left without lysing. If any chunk of the tissue is left, sonication step can be repeated. Now the next step is to centrifuge so that we can get the clear supernatant. Centrifugation can be done for 15 minutes at 8000 times gravity at 4 degrees. After centrifugation, tissue debris are settled down and clear supernatant can be collected. Now the supernatant should be collected in a clean, fresh ependraft. This supernatant has all the proteins of this tissue. So this is the collected supernatant that have all the proteins. After getting this protein sample, now they can be taken for the downstream process such as protein quantification and quality check, or they can be stored for further use.
All right, thank you Sushmita and Meher for conducting this hands-on activity. I hope participants are at least able to visualize that how simple it is, and this is one of the very efficient way of getting very high protein yield from the sample type, especially the tissue type of samples. But often you will be using other methods as well, and so I'm going to give you glimpses of some other methodologies and a very short glimpse of different type of methods which you can use. Not necessarily that the urea method can be only used for the tissue. It can also be used for the cell lines and bacterial pellets as well. But likewise, Trizol extraction method I'm going to show you with the bacterial proteomics type of workflow, but it can be used for other type of sample, including tissue as well, right? So you can try many things. I think the good thing is, if you are not the your lab has not worked in the proteomics earlier, and you have no prior background and idea for what is the best workflow for doing proteomics in your lab, it's good idea for you to first try out different type of extraction methods, see which one is the easier and also provides you high quality of the sample on the gel and provides good yield and quantification looking at the different type of quantification methods. So Trizol method is one of the method which we found quite easy and good to work with. And many of faculty participants and students who are here might have already used Trizol method for doing the RNA extraction, right? So Trizol reagent that possesses actually phenol, guanidine isothiocyanate, and L-laurosyl sarcosine, and each one of those have very distinct role and they're very important. The phenol and chloroform which is used in the method is going to dissolve the lipids and polysaccharides and going to help in the phase separation for the extraction of the nucleic acid. Guanidine isothiocyanate is going to break open the cell membranes and inhibits the ribonucleases and proteases. Therefore, it has been highly used for the RNA-based work. And the detergent, which is ionic surfactant, which we are using, that is can be used for the denaturation. So in this method, we are going to use different phases, different layers, and that will be helpful for us to do sequential extraction. So think about, you got some, clinically important sample, and from same sample you want to perform the RNA, DNA, as well as the protein-based work. And now you have very limited amount of tissue or sample available. So Trizol in that way can be very good way, because you're already assured of good quality of RNA which you will get. But from our experience, they've also got very good quality of the protein from that. So the aqueous phase, which is the chloroform phase, that contain the RNA. The interphase contains the DNA, and organic phase will have mostly the proteins. So you can actually separate these layers, and with that you'll be able to get the same sample RNA, DNA, and the protein. Let me show you briefly how this experiment can be performed.
Bacterial protein extraction. This process involves culturing of bacteria, harvesting and sonication of the obtained culture, followed by protein extraction. Bacterial culturing. Clean the laminar workspace thoroughly with ethanol and keep the master plate having the bacterial culture ready. Light the spirit lamp or Bunsen burner to maintain sterile and aseptic conditions throughout the process. Carefully remove a bacterial colony from the master plate and inoculate the autoclaved growth media. Incubate this inoculated sample at 37 degrees Celsius for 6 to 8 hours with constant shaking to allow the bacteria to grow. The turbidity of the culture, which gradually increases, indicates bacterial growth. Culture harvesting. Transfer the grown bacterial culture to a fresh tube under sterile conditions. Centrifuge this tube at 12,000 RPM for 10 minutes, maintaining a temperature of 4 degrees Celsius. Transfer the pellet obtained containing intact bacterial cells to a fresh microcentrifuge tube. Wash this pellet thoroughly with phosphate buffer to remove any unwanted debris. Sonication. The resuspended pellet is sonicated on ice to enable the bacterial cells to break open so that its contents are released. Sonication involves the use of high-energy sound waves that are capable of breaking open the outer membranes of cells. All cellular contents, including proteins of interest, leak out of this disrupted membrane. Carry out the sonication procedure for 30 seconds with a pulse of 1 second at 40% amplitude. Once it is complete, centrifuge the contents and collect the supernatant that is obtained. Protein extraction. The Trizol extraction protocol allows efficient separation Chloroform to the supernatant obtained after sonication. Mix the contents thoroughly by vortexing. Next, add chloroform to this solution.
All right, it seems there was some connectivity issue, but I also realized that we are kind of quite delaying now this session. I have a few more methodologies to talk, and I don't want to talk in between your lunch. So let me just try to conclude this session right now, where I started discussing about the need for thinking about experimental design, as well as various type of strategies in which you can prepare your sample very well, looking at questions which you want to address. Every type of sample is going to pose you different type of challenges. And we showed you some demo, some idea for taking a tissue sample and how to process that, with the urea lysis method, or you can use Trizol extraction method. But likewise, I'll show you some more sample type and how to do the good protein sample preparation, protein extraction from other biological sample as well. For instance, plasma sample, for example, cerebrospinal fluid, looking at nasopharyngeal swab, or other type of sample types as well. So I'll cover a couple of these sample type more, so that you are more exposed about thinking what are the challenges, inherent challenges involved, and how creatively you can come up with your own workflow of doing the best protein extraction method for proteomics application. Unlike what you have been doing protein extraction, which you simply just run on the SGS page to know the protein, that was slightly different than the context what we have here, because here the goal is to look for really almost all the possible protein from the given sample type, and you want to really enrich them to do the deep proteome based investigation. So that's where the strategies have to be slightly different. And also when you are thinking about how to best do that for a given type of experimental platform, then you have to think about the amount of protein requirement, the type of controls to be used beforehand, which is not possible when you are doing just the routine protein extraction for the normal electrophoretic based work. So I'll stop here for this session. I hope that has started giving you an idea that in the course, in this workshop, we are going to talk to you about many practical approaches, not just theoretical lectures, where we can give you some insight about how to prepare yourself well for performing the good proteomics based experiments. And when you think about quantitative proteomics or targeted proteomics, these are all fancy things which are, everybody wants to jump into it, but nothing is possible if you do not know how good to prepare your sample to start with, because finally the technologies are very robust, like mass spectrometers, microarrays, various type of other platforms. They're very robust, but your input sample type is not of good quality. And sometime we get a sample from a user who want to do some good proteomics work, but the the protein quality is not good, amount of peptides which is the process from that is not good. Then of course your mass spectrometers cannot provide you good quality data if you do not have the good input sample type. So please do pay attention to these type of detail. If you are working on some sample type which is challenging sample type to work with, we'll be happy to discuss, deliberate as a group how best to overcome the challenges and try to work on those kind of samples. And let's continue this interaction, and by this lecture to be continued post lunch, which we will again continue and talk to you about first other strategies of the sample preparation, followed by protein quantification and other quality control checks on SDS page. And once you are ready for these, then you can take it forward for the next step, which will be your mass spectrometry experiment which you want to perform, or 2D dye experiment which you want to perform, and how best you can do for in solution digestion or in gel digestion, so that the samples could be ready for labeling for the next steps. So I'll take a break here. We can take a pause. Please feel free to ask me any questions now, or if you want to reserve yourself and want to discuss those in the afternoon session when we have more interactive session. We can do any of these, but I hope you started probably getting a feel of it that proteomics is not just simple like doing a PCR or NGS where you can do a simple DNA extraction and the instruments will do the job for you. Whether it's the PCR, which is very robust, or NGS, both are doing a lot of stuff automatically for you. In field of proteomics, you have to be very intellectual. Think about what samples you are going to look into, how to prepare it well, what type of technology you can use to get the deep proteome analysis and best quantitative analysis, and then only you can be able to get some sort of meaningful data from your experimental strategy. That's all for this session. Thank you very much, everyone, for staying throughout the session since morning. I hope you are enjoying this workshop so far, where we have got really some eminent speakers to start with today. Dr. Surekha Jinde, Dr. Mark Wilkinson, the people who are pioneers of the proteomics, and you got some other flavor as well based on your request of giving you some technology insights as well as genomics little bit flavor. Dr. Anirban Chatterjee gave some idea for the NGS based workflows and their clinical applicability. And now we are moving into more in the workshop mode to give you idea for practical aspects of doing proteomics and how to take these workflows forward. So whatever we are doing today and tomorrow will be kind of in a theme when we are preparing the samples, and these samples will be performed for the quality control checks and then taken forward for the gel-based proteomics, and eventually we will take the same samples for the mass spectrometry based identification and quantification based workflow. That's all for this lecture and this session. Thank you very much, and we will again